Hi everyone, I'm Xu Xiaoyue from CSBL. Today the title of the journal I'm going to introduce is a bacteriocin expression platform for targeting pathogenic bacterial species. This is the article related to the targeting pathogenic bacterial species with a bacteriocin expression platform using bacterial secretion system. The article was published on Nature Communication last year. Here are the highlights of this article. Bacteriocins are known as antimicrobial peptides that are naturally produced by many bacteria. So in this study, authors developed a modular bacteriocin secretion platform that can be used to express and secrete multiple bacteriocins from non-pathogenic Escherichia coli host strains. They created anthracene A and anthracene B secreting strains that show strong antimicrobial activity against Enterococcus faecalis and Enterococcus faecium in vitro, and characterize this activity in both solid culture and liquid culture. They also developed a lot-Volterra model that can be used to capture the interactions of these competitor strains. In this study, they confirmed the simultaneous exposure to ant A and ant B can delay the growth of the Enterococcus. Microbial communities have a significant influence on human health and have been linked to a variety of disease conditions. As a result, there is increasing interest in approaches that modulate the host microbiota interaction to prevent and treat illness. One approach is to use engineering biology techniques to construct engineered live biotherapeutic products. These apps are typically bacterial strains that have been modified to perform a therapeutic purpose. Another promising application of apps is the production of antimicrobial peptides that can be used to target antibiotic-resistant pathogenic species. AMPs are a class of small peptides that display antimicrobial activity against a range of bacteria, fungi, viruses, and parasites, which have emerged as a potential alternative to traditional antibiotics in the face of concerns over antibiotic resistance. In this study, authors focus on a subset of AMPs known as bacteriocins. Bacteriocin-producing systems have been previously reported by JADAT, which repurposed the microcin V secretion system to express multiple bacteriocins, and PMPS2 platform showed that the simultaneously producing multiple bacteriocins was able to slow the development of resistance in the target species. Although the system was successful, it was shown that the operand layout of multiple bacteriocin producing strains greatly affected the antimicrobial activity of their PMPS2 platform. In this study, authors report the development of a new bacteriocin secretion modular platform that can be used to target pathogenic species based on the CDAR modular assembly standard. Modular cloning is a one-part digestion and ligation multi-part assembly method derived from Golden Gate, which also is applied in my own experiment to construct the surface display toolkit. This format allows for simple library propagation and combinatorial assembly from reusable parts with reliable ligation of multiple fragments in a one-part reaction. The overview of bacteriocin expression platform used in this study is given in this figure. Expressed bacteriocins are exported from the host strain using a secretion tag and can then target pathogenic strains in the extracellular environment. In this study, 4 common used secretion tags, PM3, OMPA, MLE, and 4A, were tested to determine if the level of antimicrobial activity differed between each tag, and the bacteriocin coding sequence linked to the secretion tag is under the control of a constitutive promoter. Enterocin A and enterocin B were chosen as candidate bacteriocins to show the efficacy of their AMP system. The antimicrobial activity of these bacteriocins against Enterococcus faecalis was confirmed via chemically synthesized peptides. They also confirmed that Ant A and Ant B did not show antimicrobial activity against E. coli cells under a limited dose. Growth curves of Enterococcus faecalis cultures grow in the presence of Ant A, Ant B, or mixture of both, show that Ant AB was able to delay the onset of Enterococcus faecalis growth at higher concentration. A previous study suggested that the proteins expressed with the PAL-B secretion signal are subsequently exported from the periplasm via the GSPD secretion pore. To confirm whether this was the case for this secretion platform, a modified version of the PM3 and A plasmid was developed, including constitutive expression of the GSPD gene. These constructs were then screened in both the GSPD knockout and the parent strain chassis. Even for strains with no GSPD expression, inhibition zones were seen, suggesting that the GSPD pore is not solely responsible for bacteriocin secretion from these strains. This will need to be explored further to identify which secretion pores are responsible for the activity seen with these engineered strains. They tested efficacy of the platform against the Enterococcus faecalis in both solid and liquid culture assays. All of the engineered strains were found to produce a zone of inhibition, which confirmed the antimicrobial activity of the engineered strain against the Enterococcus faecalis. The largest inhibition zone was found with the OMP and A strain, and the smallest with the 4A and B strain. No inhibition zone was observed for the control construct, which did not contain a bacteriocin expression plasmid. A competition assay between engineered E. coli and Enterococcus faecalis were performed in liquid culture as shown in Figure A, and as shown in Figure B, in control co-culture, Enterococcus faecalis outcompetes and excludes E. coli, but after secretion of bacteriocins by engineered E. coli strains, Enterococcus faecalis disrupts this exclusion with varying degree of efficacy with each bacteriocin. Or bacteriocin producing strains inhibited Enterococcus faecalis growth compared to the control. Moreover, Ant-A secreting strains outperformed their Ant-B counterparts, consistent with previous reports of Ant-A's superior antimicrobial potency. And a summary statistic which ranked the performance of the strain with proportion of Enterococcus faecalis at 10 hours is shown in Figure C. The authors then investigated whether co-secretion of multiple bacteriocins would enhance antimicrobial efficacy. They engineered 4 strains co-expressing Ant A and Ant B with the same secretion tags using their plasmid system, as illustrated in Figure A. As a result, the co-culture assays demonstrate that the Ant AB expressing improves the inhibition activity against Enterococcus faecalis with secretion tag of MAL-E and PM3 compared with individual Ant A or B counterparts. However, co-expressing of Ant A and B with the secretion tag OMPA and 4A did not improve on the activity shown in the single bacteriocin Ant A with tag OMPA and 4A in co-culture. This may have been due to the increased metabolic burden placed on the hosts of these constructs, which is indicated by the decreased growth rate and increased lag time of these strains. Therefore, in following experiments, authors tested with the E. coli strain which expressing Ant A with PM3 secretion tag. To show the wider application of this system, authors screened the PM3 Ant A expression plasmid in 3 additional E. coli host strains. BW25113, NB5-alpha, and NISO1917. Enterococcus faecalis inhibition zones were seen at similar levels for all host strains as shown in Figure A. In addition, this system also showed antimicrobial activity against a very isolate of Enterococcus faecium as well. Then authors used mechanistic modeling and Bayesian statistics in order to gain further insights into the dynamics of the bacteriocin interaction. They tested a number of pairwise interaction models and compared by fitting the data from the PM3 system, including monocultures of Enterococcus faecalis and E. coli single and geobacteriocin production stage in co-culture with Enterococcus faecalis. A model containing a saturated local Volterratum for bacteriocin interaction was the best model. Under typical in vivo conditions, other strains of the host microbiota will be present in the environment, alongside the engineered and the target strains. Therefore, authors further tested the activity of the bacteriocin-producing alps in a 3-strain community. As the third strain, they used a GFP fluorescent E. coli NEB5-alpha strain, referred to here as the bystander species. In a 2-strain co-culture, Enterococcus faecalis was able to outgrow the bystander strain. This was also the case in the 3-strain co-culture with the control. In contrast, in the 3-strain co-culture with the PM3-A, Enterococcus faecalis growth was suppressed and the bystander strain was able to dominate the measured OD. They suggest that the PM3-A alps strain is able to suppress growth of the target strain, creating a niche that the bystander strain is able to exploit. To date, they have only screened AB co-expressing strains that use the same secretion signal. Therefore, authors performed a screen of PM3-A expressing construct with multiple promoters and RBS parts to demonstrate multiplex reaction and created strains that use a mixture of secretion signals. From the multiple reaction, they were able to produce alpha strains that showed a range of antimicrobial activity. However, even in co-culture, the conditions found within the human gut will be extremely different to those encountered in vitro. Therefore, in vivo testing or improved in vitro methods will be required to more accurately predict final strain performance. In conclusion, in this study, authors built a modular AMP production platform based on the CEDA-MOCLO standard and chose 4 set pathway tags to test the secretion of bacteriocins, and also engineered a PM3-A construct with and without constitutive GSPD expression, observed that bacteriocin export does not solely rely on the GSPD pool. Then, authors developed co-culture assays to mimic in vivo delivery contexts where engineered and target strains grow in proximity. And the result indicated that PM3-A alpha strain is able to suppress growth of the target strain. In addition, co-expression of ANT-A and ANT-B improved Enterococcus faecalis suppression in some secretion tag like MEL-E and PM3, but imposed metabolic burdens in others like OMP-A and 4A. Furthermore, authors used mathematical modeling and Bayesian statistics to gain insight into the dynamics using the co-culture data. Finally, authors demonstrated multiplexed promoter RBS screens in the PM3-A background to generate strains spanning a spectrum of antimicrobial activities. This is the end of my presentation. Thank you for your listening.